So why I'm saying this, say like when a patient comes to the clinic, the doctor sometimes get confused how to treat them. And they send, parallelly we are in the lab, we are also working with the patient, with the biospecimens. But if we could make something, some diagnostic kit, some drug, which actually help the patient, then the doctor is also happy and the patient is also happy. And we are, most importantly, we're using public money. We're using taxpayers' money. So if a part of our research, even a percentage of our research come to the clinic, could help the patients, so nothing is better than that. So that's all what I'm going to say. And actually, you have seen during COVID, a lot of diagnostic kits were made. So actually, we were all the national institutes, they came forward using their real-time PCR machines. They did those tests. So then we realized there is a gap, and that gap can be bridged. So it's all about to come forward with a good intention, and then definitely it's possible, because the common intention is the betterment of the patients, the management of the disease. So the location is different. We are doing at a different place, and the doctors, they're directly involved. So the research questions should revolve around this, how the bench should come to bedside. So again, the same thing I'm reiterating, the doctors and us, the scientists, and there should be somebody, some technical person, because to make those kits, we don't have the expertise. We work on cell lines, we are biochemists, cell biologists, molecular biologists. But some transformation is required to bring those discoveries to the clinic, and we need some technical help. So that's why in IITs, a lot of those biomedical engineering departments are being set up. So they act as a bridge between us. And obviously, we need these kind of people to fund us. So without that, nothing is impossible. It could be government, it could be private funding, because we don't see much of private funding in our country, but in other countries, this is very much prominent. And then at the end, it's all about better management of the disease. So now coming back to the biomarker, what are they? And now you understand the discovery of biomarker is huge translational importance. So what is translational research? Translational research is, it's not like this translation is not the central dogma, replication, transcription, translation. So this is the research which is being done, is being translated to the society. It's called translational research. Whether the effect of the research, whether the benefit of the research is transmitted to the society or not. Okay. So the next few slides I'll just cover what is biomarker, because the conceptual thing is important before we move on. Different type of biomarkers. Why do you need? দাদা ঠিক ছিল তো শুনতে পাচ্ছ না? আরো ইকো হচ্ছে, মাইক বন্ধ ছিল তো, বাট তাতেও তো অসুবিধা হচ্ছে না, ঠিক আছে, কারণ ইকো হচ্ছে। এনিওয়ে, থ্যাংক ইউ। So why do you need them? And biomarkers in the context of pancreatic cancer. So now you have understood from Professor Pandey's introduction that my lab works mainly on pancreatic diseases. So I chose pancreatic cancer as an example, disease example, because then I can at least share some of our recent works also with you. So I'll just read out this thing. So this is a definition which was coined by National Institute of Health, USA. So what is biomarker or biological marker? So the objective indications of medical state observed from outside of a patient, which can be measured accurately and reproducibly. So that means for a biomarker measured in a patient in USA or in Pakistan or in Uganda should not vary much. So they should be accurately and reproducibly measured. So a molecule, object or indications or medical state that should be observed from outside of the patient. And there are couple of different subtypes of biomarkers. There are predisposition biomarker, diagnostic biomarker, prognostic biomarker, and predictive biomarker. I will come to all of these. First is predisposition biomarker. So what is predisposition? So a genetic predisposition, or also called a genetic susceptibility, is an increased likelihood of developing a health disorder based on a presence of particular biomarker. So describing it in easy language, so this tells whether a particular person is susceptible for any kind of disease. So just remember the word, it's not a patient, it's among the population whether an individual is susceptible for a particular disease or particular condition. So we've all heard about Angelina Jolie. So she had the breast cancer gene, the BRCA1 mutation running in her family. So then she underwent mastectomy and ovarectomy. So this is predisposition. So she knew in future she might have breast cancer or ovarian cancer. So identification of these persons who are predisposed to any kind of disease, be it cancer or any kind of disease, is important because beforehand they know. So another common example is APC gene mutation in colon cancer. So there is like a hereditary condition which leads to colon cancer, and the percentage is very high. So hope I could explain what is predisposition biomarker and the importance. Okay, this is mainly done in the population. Next is diagnostic biomarker. This is mostly used. What is this? Is used to confirm that a patient has a particular health disorder. Now, it's not an individual, it's a patient. So, doctors suspect that somebody has a diseased condition. Now, to identify whether the disease is already set in or not, this is confirmatory. So, the indications of a person say that he might have diabetes. Wounds are not healing. He is feeling early onset of fatigue. So then doctor prescribe a blood glucose levels. That's a diagnostic biomarker. So this is, suspects the patient and then confirms. These diagnostic biomarkers, they confirm whether the person has disease or not. Then comes prognostic biomarkers. So prognosis, what is the meaning of prognosis? The progression of a disease. So these type of biomarkers, they say whether a patient will have a better prognosis, whether the person having the disease will be soon recovered or whether it will be difficult for him to recover. So how the disease will progress, whether the cancer will metastasize to other organs or not. So these are called the prognostic biomarker, the disease prognosis. And the last one is predictive biomarker. So there is not much difference between prognostic or predictive. And so predictive biomarker is sometimes linked to the response to the drug. So if a chemotherapeutic drug is given to a patient, so how does this drug perform? They block some pathways. I'll come in details in later slides. They block some pathways. They block some genes so that the cancer cells do not proliferate. So that gene mutation varies from individual to individual. So that means the response of that patient to the chemotherapeutic drug, that's also individual dependent. So these predictive biomarkers, they predict whether an individual is responsive to that particular drug. So hope I could explain like the difference between different biomarkers. Now the major task is, how should you know? So then the rest of my talk will be different techniques which are used to detect whether an individual is responsive to a disease or whether we can distinguish a patient or a normal healthy individual or not, right? So now we can even think this kind of things just with the advent of different technologies. So in 15-20 years back, it was almost impossible. We can ask now more and more difficult questions. Okay, so as I said, we have covered this part. The last part is different type of biomarkers based on these next generation omics driven technologies with pancreatic cancer as an example. So that's why you need an introduction of pancreatic cancer. What is pancreatic cancer and what are the difficulties? So pancreas, you know, is an organ very deeply located in our abdomen, and this is just beneath the liver, and this part is the duodenum, and this is a pancreas, and I think there are slides, yes. So if we just look at the pancreas, it can be divided into multiple portions. So this part is the head of the pancreas. So if we go back again, so this is the head of the pancreas, which is adjacent to duodenum. So I think this kind of this little bit anatomy you all remember. This is here is the stomach, and this is the duodenum, and here in this groove the pancreas resides, and one duct, this is the bile duct, and there is hepatic duct. They form a common bile duct and they merge with pancreatic duct and open in the duodenum. So liver, the bile duct brings bile, pancreatic duct brings the pancreatic enzymes, and they open into the duodenum where semi-digested food from stomach comes in. So the rest of the digestion takes place in the small intestine. And pancreatic enzymes, you all know, they have like lipase, they have protease, they have amylase to digest all different types of food components. So as I said, this is the head of the pancreas, this is the neck and body, and this is called the tail of the pancreas, and this is called the uncinate region. So mostly the tumor, the malignant tumor, they form in the head region of the pancreas, say about 85 to 90%. So this is actually a resected pancreas with the mass or a malignant mass in the head. So if you again go back, this resected part is like this portion. So this much of pancreas is like, kept inside the patient. This is actually one of the tumor tissue collected by my students. They had took a picture. And the surgery of the pancreas is very morbid. Surgery of the pancreatic cancer is very morbid. So what do they do? If you follow the picture, this is post-surgery and this is pre-surgery. And the gray part, the discolored part, those part is removed. So what is removed? The gallbladder, just remember the mass is here in the head part of the pancreas. So the gallbladder is removed, bile duct is removed, even part of stomach is removed. This much portion of the pancreas is removed, only the tail portion remains, and the entire duodenum is removed, part of jejunum is removed. is about a 6 to 8 hours surgery, and not everybody can do that. Needs special kind of expertise. In Calcutta, maybe there are 4 or 5 surgeons who perform that. And what after that? So remove the cardia. The next is anastomosis, the joining, because the patient has to survive. So what do? The remaining part of the pancreas, they join with jejunum. It's called pancreatojejunostomy. Pancreas being joined to jejunum. Remaining part of the stomach again joins another, they make another hole in the jejunum and joins over here. It's called gastricojejunostomy. So that means, and the whatever we eat, they come directly, it's a semi-digested food, come directly to jejunum, and pancreas also secrete all the enzymes now directly in the jejunum. So you understand the patient is heavily compromised, but at least they could survive. Yeah, they could survive. And this is the resected part. If you follow this gray part with this one, this is the gallbladder, this is the duodenum, and this is the pancreas, head of the pancreas, and this is some part of the stomach. So huge surgery is called a Whipple's surgery, based on a US surgeon, Dr. Whipple's. He actually invented the did the surgery. So pancreatic adenocarcinoma. So most among different type of pancreatic cancers, pancreatic adenocarcinoma is actually 80% of pancreatic tumors, and it's most common GI malignancy after colon cancer. And the least 5-year survival rate of 3%. So this has recently increased to 8 or 9%. So what does it mean? 5-year survival rate of 8% or 9%. What does it mean? That means if today 100 individuals are diagnosed with pancreatic cancer, after 5 years, only 8 patients will survive. So 92 of them will die within 5 years. So that itself tells how aggressive the disease is.
So and what about post-surgery?
Huh?
What about post-surgery?
What about post-surgery? So post-surgery, the post-surgery complications are there. So even after removal, so I'll come to some of the points. You'll understand why surgery can extend life maybe few years, not more than that. The most important is a very pertinent question. And yeah, as I said, it's like a very aggressive tumor. But this is the most important problem. Why? It's clinically quiescent for a long time and hence presented in very advanced stage. And another problem is this organ is located deep in the abdomen. So I think many of you have experienced, say, our family physician, they oftentimes check the liver and could say, okay, liver bereche. They could feel the expansion of the liver, the enlargement of the liver. But pancreas, they cannot feel. So it's most of the time the disease, the early onset of the disease is undetected. And yeah, so this is, I'll come to another problem later, I think, when the slide comes. So in continuation with the previous slide, so it's a very aggressive disease, and there's some statistics showing the huge number of onset in different countries. And when you look at the leading cause of cancer, death, so pancreatic cancer might not be the cancer which is very much predominant, but in terms of death, it's very high because of the issues I was talking about, its aggressiveness. And yeah, this was the slide I was mentioning about the challenges in early diagnosis. Why it is presented at late stage? What are the challenges? There are no symptoms in early stage of the disease. The symptoms are, say, like weakness, abdominal discomfort, loss of appetite. So what happens? Firstly, self-medication. Many people, they start self-medication at home, thinking that, okay, I'll be fine. Those antacids, now those Gintac, Rantac, omics, self-medication. It doesn't work. 15 days, 1 month. Goes to the family physician. Family physician again starts the same thing. Antibiotic, nothing works, but another month is gone. Then family physician also doesn't want to refer because there's sometimes there is ego problem, like I can't treat it. Do ultrasonography. Ultrasonography also cannot pick up all these things, but the problem persists. 2.5 months gone. Then he is referred to a gastroenterologist. Getting appointment, getting a good gastroenterologist, getting appointment, moving to a big city, and the gastroenterologist often doesn't believe in all the tests done by the suburbs or village doctor. He will again repeat all the tests and all these things. 4.5, 5 months gone. Then also the problem persists. Finally, they recommend CT or MRI at the 6th month. Then it is diagnosed. So now gastroenterologist cannot do anything, refers to a surgeon. So with all these things, the cancer already spreads. So it's a very aggressive cancer. And if you wait for 6 months or more, just nothing can be done. So when we get the tissues, the operable tissues, they are all like stage 2A or stage 2B. But this is only 20%. I think one of the slides it had. It's about 20% of the pancreatic cancer patients, they come to the clinic. It's like operable cases. But mostly the doctors open the abdomen and see it has metastasized to multiple organs in the peritoneum, liver. Those are the two major sites, liver and peritoneum. And then they had to close. So my students, they go and wait outside the OT. Most of the time, the doctors come and say, no, we cannot give tissues because they're inoperable. So the only option then is the radiotherapy or chemotherapy. So this is a huge problem. And the existing biomarkers, this is one of the biomarker, blood-based biomarkers, is carcinoembryonic antigen CA19. It is a glycosylated protein. So the problem of these biomarkers is this is not specific to pancreatic cancer, neither sensitive to pancreatic cancer. So any gastrointestinal disease, GI cancer, it will be elevated. So measuring this, it cannot be said that the patient has pancreatic cancer. So like the existing CEA, carcinoembryonic antigen, CA19-9 and CA242, if you look at the sensitivity and specificity, none of them have good more than 90% sensitivity or more than 90% specificity. For a biomarker to be effective, both the sensitivity and specificity had to be very high. So the early detection of the disease is a challenge, and there is a need for early detection biomarkers. So how different research from ages tried to develop different type of biomarkers for pancreatic cancer. So these are called, as I said, these three other traditional biomarkers, but now we are expanding to metabolites, cell-free DNA, autoantibodies, cell-free non-coding RNAs, inflammatory factors or growth factors, circulating tumor cells, and exosomal cargo. So I'll explain most of these. So there are a lot of jargons and terms. So these are all based on next generation omics technologies. So earlier it was not possible for us to even think of these things. So detection is later. So now the term is molecular diagnostics. So we have heard of diagnostics. Now it's molecular diagnostics. What is that? It's a collection of techniques used to analyze biological markers in the genome or proteome, that's in the individual's genetic code, and how their cells express their genes and proteins by applying molecular biology to medical testing. And again, the genomics. So this is actually the central dogma. I don't know, maybe it's not visible from clearly from back. So DNA to RNA, protein, and then here it's written is sugar, nucleotide, amino acid, and lipids, right? So when we say genomics, take genomic techniques, that's something in DNA. Transcriptomic techniques that finds out what are the changes or alterations in RNA. Similarly, proteomes and metabolomes also, which actually measures any differences in sugar level, sugar, nucleotide, amino acid, and lipidomics is very much prominent now. So a lot of studies are being done on lipidomics. So the specific lipid alterations between individuals. So parallelly, another term is circulating tumor cells and what is actually called the liquid biopsy. What is that? So we now know the tumor cells, when in the primary site, how do they metastasize? So our research for decades has told us there are different steps of metastasis. So the tumor cell, there is in the cell itself, there is epithelial to mesenchymal transition. I think Dr. Ganguly is going to elaborate on that. His talk title has all these terms. So epithelial to mesenchymal transition. So unless a cancer cell is transformed to the or attains the mesenchymal properties, it cannot start the dissemination process. So from the primary tumor, it then traverses through the bloodstream and then travel to a different organ. For pancreatic cancer, it's like mainly liver and lungs, and also obviously the peritoneum, and then forms a secondary lesion over there. So that means the tumor cell with all these mutations are traveling through the bloodstream. Imagine a situation if we now could detect the tumor cell before it has gone to a secondary organ and like homing to a different organ and start metastasis. If we could detect the tumor cell based on the specific mutation it is carrying for the specific type of cancer. So, am I making sense? So, I'm repeating once again, the tumor cell for pancreatic cancer has a specific mutation that we discover from our genomic technique. So, in pancreatic cancer, it's a KRAS gene, 12th codon of KRAS gene. If somebody has mutation, it's obvious, it will develop pancreatic cancer. So, this primary tumor having KRAS 12 codon mutation will be shed into the bloodstream of the individual. Now, if we isolate or if we take out that particular, detect that particular tumor cell, that is circulating tumor cell, and identify the KRAS mutation F12 position, then we can say the individual has started, the tumor of the individual has started disseminating the cells. So this is how the technique works. It's called the liquid biopsy. So biopsy is what? We take tissues from the primary tumor or the secondary lesion. In liquid biopsy, we are taking samples. We are sampling from the blood and detecting the tumor cell which is traveling in the blood, in the liquid condition. That's how it's called liquid biopsy. You'll listen to these kind of jargons quite often. So now, genomic exploration, what I just said. So we collect blood and I don't know whether it is visible or not. Here it's written CTC, circulating tumor cell enrichment. So there are now methods. We have developed techniques to enrich. When you isolate blood, there will be so many blood cells. The tumor cells will be hiding over there, like handful of tumor cells among all these blood cells. So there are techniques to enrich those circulating tumor cells, because the next step will be what? These DNA extraction, because we are looking for specific mutation pertinent to pancreatic cancer. So unless we enrich those circulating tumor cells, we are not going to have enough amount of DNA even to detect with our next generation technologies. So this is important. Enrichment of the CTCs and then DNA extraction, and the normal, if you have targeted, if you know, okay, I am going to detect the KRAS 12th codon mutation, you do a PCR. Simple. Or if you want to detect, okay, not only that, I would like to explore other mutations also in other genes, then you use sequencing. That will cover the genome-wide changes, not only 12th codon mutation of KRAS. This is more logical, to be sanguine, to be more like in a confirmatory test. Similarly, from the same blood, some of the cells will be ruptured. Some of these circulating cells will be ruptured, and that will be while traveling through blood, and that will shed their DNA, genomic DNA, into the bloodstream. So that's called circulating tumor DNA, ctDNA. This is CT, C's circulating tumor cells, and ctDNA is circulating tumor DNA. The cells have ruptured while traveling and the DNA is shed into the bloodstream. So there is no need of enrichment of circulating tumor cells now, because all we want is the DNA from them. So DNA is already traveling in the blood, and we can do the same thing, either do PCR or do the sequencing to identify specific changes, specific mutations, what we expect to be there in pancreatic cancer. Because we want to detect whether the person is developing metastasis, whether the person is developing cancer. So we know we are going to expect mutations in these genes. So if we get that, so then we can confirm that person is having the thing and it has started disseminating. So just an example of what are the different mutations in different pathways or genes in which has already been reported to pancreatic cancer on using these kind of techniques. Say like this chromatin remodeler genes, Wnt signaling pathway, Notch signaling pathway, TGF-beta signaling pathway. So this cell cycle, DNA repair. So these are the genes involved in these particular pathways, and mutations have been identified over there. So why it is important? Once we identify the mutation and say that the person who has come to the clinic has mutation for, say, like mTOR signaling pathway. He is not having mutation for Notch signaling pathway. So then we can design drugs which can block the overactivation of the pathway. Most of the time, these mutations are hyperactivating mutations. So they overexpress those pathways so that the cells proliferate. So if we devise some drugs, some inhibitor molecules, and stop the hyperactivity of that particular signaling pathway, then automatically some amount of braking in the progress of the disease is possible. But the importance is, what I want to highlight is, what is the importance of genomic technique in these is not everybody is having mutation in all the genes. Not all the patients, 10 cancer patients, they're coming to clinic, they're having mutations in multiple genes. So earlier, what we used to do 25, 30 years back, there is a single size for everybody. Single size search for all of them. So now we know it's not the case. The disease at the molecular level, that's different. Each one is having mutation in different genes, but the outcome is the same disease, is the proliferation of the pancreatic cancer cells. So that means we cannot target all 10 differently. So most of the time, so now I think it's very much in the clinic also. So when somebody has cancer, they first do the profiling, the genetic profiling, do a NGS based mutation screening. They have panels based on previous research work. Okay, for breast cancer, these are the common mutations. For pancreatic cancer, for colon cancer, these are the common mutations. They see how many of those the individual carry, and based on that, what drug can be given. It could be that one person comes to the clinic with one particular mutation. For that, there is no drug. Nothing can be done. But one fortunate person have mutation in one gene that can be targeted with the existing drug. So what people are doing now, they're identifying first what are the mutations present over there, and then whether all of them can be targeted, because targeting can be done individual level, and there's a huge collaboration between, it's an interdisciplinary work. The biologists come, chemists, and then structural biologists. It's a huge interdisciplinary work. So similarly, then we come to the next part, that's a transcriptomic based detection. So we completed the genomics part. When you talk about transcriptome, there are coding and non-coding transcriptome. So coding means that produces mRNA, and mRNA produces protein. And the non-coding means there are microRNA. You all have seen the last Nobel Prize was on microRNA. And then there are long non-coding RNAs. So these are all actually the fine-tuning of gene expression. They do the fine-tuning of gene expression. They are regulatory RNA molecules. So I'm not going to details of the generations, how microRNAs are produced. So I'll just keep this slide. So this is important because we have some vesicles in the cells. Those are called the exosomal vesicles. What are they? So the cells, they secrete those vesicles, they're pinching off from the cells. It could be tumor cell, it could be normal cell also. And they are released in the bloodstream. And these cells, they contain cargoes. What are cargoes? These are vesicles. Why they are called cargoes? Because they're some packets of the lipid layered packets being secreted by cells. And they have proteins and RNAs in it. Specific proteins and RNAs in it. It's not like a random. So what happens when a tumor cell, so tumor cell also secretes exosomes and they travel through the blood. And it has been shown that the exosomal vesicles secreted from tumor cells, they contain specific microRNAs which are upregulated within the tumor cell. So that means they're like characteristic. So again, from tumor cell we are coming to the blood. Just remember what we have just told during CTC isolation. So just take the blood, because the most important part for biomarker discovery is it has to be non-invasive. And if you remember the terminology, has to be monitored from outside. So once we have come to the blood and we could detect the exosomes carrying specific microRNAs characteristic to the pancreatic cancer. So then a person can be similarly detected for the disease, either disease diagnosis. So if we start screening the individuals susceptible to pancreatic cancer and see when a particular microRNA starts appearing in their blood. So as soon as you detect, even the person doesn't have symptoms, we'll see those molecules coming in the blood. So this is the beauty of these kind of techniques. So again, so this is just the method how they are detected. Once the exosomes are isolated, the small RNAs are isolated, and we can do qRT-PCR or small RNA sequencing. These are the next generation techniques. Again, omics based techniques, small RNA sequencing. They discover all the small RNAs. They detect all the small RNAs between the individuals and then compare which is more in one person, which is less in one person. So this is just a contribution from our lab. We had published some time back this particular paper where we have identified a set of say like 8 or 10 microRNAs, the bold ones, in pancreatic cancer patients as opposed to the normal healthy individual.
So what would be the cost of this kind of test?
Cost?
Yes. Is it feasible or not?
Yeah, so these microRNA based test has not yet come to the clinic. So it's still at the research step, and the cost is obviously, I mean, it has not come to the market and it is a little bit expensive. So we are yet to find out whether
Will come to the market.
yeah, people are trying heavily whether they could be brought in the market in future, because this is definitely a change which is being observed in these microRNAs, but actually there are 6,000 different microRNAs, and that varies. The presence of these microRNAs varies from, like, say, ethnicity. What we see in Indian patients is actually not matching to Westerns. So there are a lot of variation in results. We are yet to, we means like the researchers, they are yet to pinpoint what could be the marker for, like, say, 6 signature markers which could be used as in the clinic. But definitely in, say, 5 to 7 years time, it should come in the clinic, definitely. So similarly, another non-coding RNA, long non-coding RNA, but I'm not going in details. So, but the concept is more important. Similarly, as you see in, you have seen in case of microRNAs, the exosomes can also contain long non-coding RNAs, which could be similarly detected. The basic theme is same, how we detect. So proteomics based biomarker, similarly, this is DNA methylation biomarker, we missed that. So what is DNA methylation? So there are methyl group added in our DNA which actually regulates the transcription of a gene. So when we say a gene is expressed, that means more and more RNA is being made by the process of transcription, and methylation group added to DNA promoter of that particular gene, that regulates whether the transcription will be more or not. And people have seen, this is again in the research lab, in the bench, not has come to the clinic yet. So people have found that there is variation in DNA methylation. So which promoter is methylated, there is variation. So it might be that in pancreatic cancer patients, particular gene promoter will have more methylation. And remember that all these DNA are being shed either in circulating tumor cells or in the blood. And we can detect the specific methylation also, whether not only gene mutation, but whether a particular promoter methylation is more or not in an individual. That also can be detected.
Promoter methylation, but there may be epigenetic methylation.
Yeah, so this is called like the epigenetic modification, this promoter methylation. Because histone modification.
If it is epigenetic, that means environmental factors.
Environmental factors, yeah.
Eating habits and everything.
Yeah, that might contribute to these. And the ultimate result is the methylation. But the histone modifications you cannot detect in blood, but DNA modifications can be detected in blood. So these also DNA methylation can be. So now coming to proteomics, so huge amount of opportunity with the proteomics, because if you think any body fluid, you can think about blood. Now we are talking about, so far we are talking about blood, DNA in blood, exosomes in blood, but proteins can be detected in other body fluids also, pancreatic juice, urine, cyst fluid. So there are sometimes cysts associated in the pancreas along with the tumor. So cyst fluid can be, so fine needle aspiration, FNSC, what do you say, fine needle aspiration cytology. So those cyst fluid can be aspirated, and then specific proteins, presence of specific proteins can be evaluated over there. And same using high-tech technologies like mass spec and all these things. So now it's like every institute has this kind of things. And okay, and lastly there is like metabolomics. Okay, yeah, sorry, here's the metabolomics. So as I was saying, like different carbohydrate molecules, specific glycosylations, specific lipids, why this particular short chain fatty acid is high in case of pancreatic cancer patients, and whether they can be detected. So these are the different techniques, omics based techniques, which are used in identification. So I'll just give last few slides. I will describe what we do. So as now you know that we work on different pancreatic diseases. So the chronic pancreatitis is a pre-malignant condition of pancreatic cancer, where there is a chronic inflammation. When you talk about hepatitis, we're all about hepatitis, esophagitis. So there's inflammation of liver, inflammation of esophagus, and that leads to chronic inflammation, leads to cancer, pancreatic ductal adenocarcinoma, and there are like other type of pancreatic neoplasms also, that's called pancreatic cystic neoplasms. So why chronic pancreatitis is morbid? Because it has progressive inflammation, which actually causes all the normal pancreatic cells to die. Pancreas is important for our digestion. It's important for controlling diabetes, because the exocrine cells, they secrete enzymes, and the endocrine cells, they secrete insulin. When the pancreatic cells are dead, then all the cells are gone. So neither it produces enzymes nor it produces insulin. So chronic pancreatitis is again very debilitating disease. So one of my PhD students, she tried to find out whether we could detect differences in benign and malignant mass. Benign mass is chronic pancreatitis, and malignant mass is pancreatic cancer, between using this gene expression signature. So what we did is collected tissues and did a transcriptome profiling, and then identified, actually I'm just skipping these, identified a 5 gene signature which could detect the malignant tumor significantly as opposed to the benign one. So these 5 gene signature, this is the ROC analysis, many of you are know these kind of things. So this is actually identifying the sensitivity and specificity of a biomarker. We'll have to do these kind of analysis, ROC analysis. So the area under the curve which is measured, it's like more than, if it is more than 90%, it's said that it has. So ROC is receiver operating characteristics. So actually when sensitivity and specificity of a biomarker, it's a statistical test. When a marker is called sensitive, so that means there are multiple disease conditions, but it is highly precisely detecting one disease. That's sensitive. This is a purely statistical test. And specific is, there are like in colon cancer, in stomach cancer, the changes are there, but specific to pancreatic cancer. So there are like, based on the gene expression data, in a population. This is calculated,
All these are co-expressed, no?
co-expressed. Yeah, so this is like together. No, this is individual expression, and if you see the co-expression, all 5 genes together, the area under the curve. So this area under the curve, how much percentage of the area of the box is under the curve, that shows the better. So the better the sensitive. So together they act as a signature. So as I said, these 5 genes we found, like as a possible biomarker, and now you are actually taking that forward to the patient blood. So what I conclude is that there are coding and non-coding RNA based technologies we are trying to explore to detect pancreatic cancer from chronic pancreatitis, and their biomarker potential is being evaluated. Yeah, this is the 2 publications. So the last few slides, this is actually our institute, and we have a shimul gach in front, and this is taken just during the spring. There are multiple of shimul gach, but this is very strategically located, this particular tree. And thank you. And we have all the facilities, whatever techniques I told, we have everything. Genomics, transcriptomics, proteomics, metabolomics, and these are quite standard. Cell culture and stem cell facility. Recently we have introduced a BSL-3 facility for working on live active pathogens, TB and SARS-CoV-2. We have started working on long COVID also. So actually this is a robot. It itself makes a library and all the sequencing, the reactions related to the sequencing. And these are the next gen sequencers. So other facilities, general labs, confocal, and this is a computational facility, because you understand we generate lot of data, omics data, like terabytes of omics data, and that has to be stored and analyzed. So yeah, there's a data center and computation, and it's a zebrafish facility. So you have an animal house for mice and hamster, and also a zebrafish. These are all central facilities. Yeah, these are all central facilities. And this is a campus in one of those monsoon days. It's a faculty housing, hostel, canteen, and this is a guest house. And we have a very nice lake. This is not a river, this is a lake, huge lake. It is a water tank. Yeah, it's a water tank, and there's a lake. It belongs to Department of Fisheries. It's a huge lake. So people mistakes it with a river. And this is my group. We call ourselves Molecular Gastroenterology Laboratory. This is my Twitter handle of my lab, and these are my current lab members. Yeah, so they are Shati and Shushmita. They are working in a project, but others, they're my PhD students. We are funded by these organizations, and definitely I acknowledge my director, Dr. Sagar Sengupta, and they are my collaborators. And Shamshiddi and Nidan, we collaborate internally, and we have also EMS Kalyani in our vicinity. We collaborate with them also. Thank you for your patience. Questions or clarifications? Whatever, yeah. Please.
Firstly, I like the lecture.
Push.
Thank you for your lecture. So first, you said that the problem with this pancreatic cancer is that detection takes a lot of time. So it is detected at a late stage. And all of these markers that you have talked about, that do they detect early?
Yeah, so that is the whole idea. I mean, if we could detect early, because we cannot, without these genomic techniques, we cannot even think of detecting those circulating tumor cells. So now we know there are techniques which is capable of detecting. Why don't we explore that in case of pancreatic cancer? That's why globally people are doing that, and there are signatures coming up, but as Madam said, that's not yet into the clinic, but definitely.
Another one, sir. It's a general question, sir.
Sure, sure.
Sir, can you talk a little bit about the vaccine that we have here for cancer?
Cancer vaccines, at least for pancreatic cancer, there is no vaccine. Cervical cancer, there is vaccine. Yeah, cervical cancer, there is vaccine, because the reason is it's driven by a virus. Because cervical cancer is the 100% the cause is a virus. Without the human papilloma virus, cervical cancer doesn't happen. So that's why we can think of a protective vaccine. But this is actually mutation based, mutation driven.
What is the general age when this develops?
Pancreatic cancer.
Yeah, it's not congenital.
No, this is not congenital or hereditary. There is a small part of hereditary thing is there. Yeah, not even hereditary. So pancreatic cancer is not hereditary. So
Definitely, yeah, that's what it is.
food habit report, recently we are seeing that with all these junk food and like unhealthy foods, doctors say that it might have some effect.
Yes, I have the same question. Does it have any relation with the fatty liver and this thing? Because fatty liver is so
Fatty
common now.
liver, not only fatty liver, there is a very hot thing coming up, that's fatty pancreas.
Yeah, because
Fatty pancreas.
because the patient is suffering from NASH or has been diagnosed with
Absolutely.
NASH, there is a chance of affecting the pancreas.
Absolutely. So, very recent thought. So, there is a huge pancreatic research group, mostly clinicians. So, based on, you have heard of AIG Hyderabad, Asian Institute of Gastroenterology, Dr. Nageshwar Reddy. So, he had conducted a meeting 3 months back. So, in that meeting, lot of people, there is a session on fatty pancreas. So, what is being seen now? Fat is being deposited in the pancreas because all these, so you have fat from pizza directly going into the pancreas. So, and that creates a niche.
I got a report, one of my relatives has been diagnosed with NASH, and there was another point written, also suffering from exogenous pancreas or something like
Now,
that.
exogenous fat accumulated in the pancreas.
Yes.
Exactly.
So
That's fatty pancreas.
yes. So what's the prognosis?
Prognosis is very bad. So, it's believed that fatty pancreas creates an environment which promotes pancreatic cancer. So, fatty pancreas is a little bit early stage.
Dietary modification can be
Absolutely, absolutely. It's not about thinking.
with genetic, certain genetic predisposition.
No, so genetic predisposition is also there. Like, because two person eating similar kind of fatty food, not having the similar fat division. So I have my cholesterol level 350. You cannot, seeing me, you cannot say, yeah. But this is the thing. So the way a person metabolizes things is based on its genetics. But apart from that,
On this question, because I have also got through the blood reports,
Mhm.
the other physiological condition and the blood pathological reports were normal.
yeah.
They didn't show high triglycerides or anything
Okay,
like that. So
okay.
all these things were normal. The person was not diabetic even.
Okay.
So everything is under control. The problem is with that the person has been was being diagnosed with
NASH and infant. Yeah, so it's understandable that when he has NASH, then degree of NASH is important. So how, whether it is
It's about F0 to F that one
Okay.
that range.
Okay, so it's understandable for NASH and similar non-alcoholic fatty liver disease and fatty pancreatic disease. But on no triglyceride level, normal triglyceride level, a little bit difficult, but definitely something has happened. Otherwise, like over the years, it doesn't have single day now. Over the years, and diet is hugely contributing. I mean, there's only, so when you are eating junk, you expect junk to happen inside. It's simple as
So
that.
to detect that, I was thinking if you do sorry.
Skin. The junk food affects everything. Affects everything.
Looking at the recent trends of increasing rates in cancer, Do we have any data regarding that molecular clock? Like we have studied না
circadian clock.
mutation. এখন তো আমরা দেখছি এত all of a sudden এত mutation. So what is happening actually? Do we have some data?
No, so there are two things.
Has that trend?
So two things. One is we are also torturing us. We are also torturing us with all these junk things and then environmental pollution. That's definitely creating more and more mutation. One part. The other part is we are detecting more and more mutations because of our techniques. So earlier, even if that were there, we are not able to detect. So, but definitely they're increasing, but we are detecting more and more.
Is there any age group kind of a thing like
For?
this detection like pancreatic cancer, like prostate and all?
Yeah, no. There is no PET now. Pancreatic cancer.
No, but
We collect patients only above 18, but even in 2025 we have chronic pancreatitis is like even 12, 14, 15-year patients. Nothing is there in the pancreas. Puro pancreas just like fibrosed necrosis হয়ে গেছে.
Data যে আজ থেকে 20 years back
20 years back,
food habit was different.
huh?
Now the food habits have changed. What is the statistics of pancreatic cancer at that 20
Yeah,
years back?
so-
What was frequency?
Absolutely.
This because-
In India, because you can, in US or Western countries, food habit was bad in 20 years back also. Yeah. So in India, we can have this transition, but unfortunately, in India, we don't have that kind of epidemiological data because clinicians, unfortunately, not many clinicians are interested in. But there are reports. Some interested clinicians.
What I'm
It
saying
has
is
increased.
another
It has
thing here.
increased.
What I'm saying is anybody studying about the microbiota and
Absolutely. I'm coming to that. This is a very interesting question and ওরা discussion. Yeah. So
skin evaluation.
yeah, sure.
বলছি ডাইরেক্ট ড্রাগ থেরাপি যেটা আছে আর ওর সাথে যে এক্সোসোম মিডিয়েটেড ড্রাগ ডেলিভারি ওটা
না exosome drug delivery না ওটা হচ্ছে exosome is actually for detection.
আচ্ছা মানে ড্রাগ ডেলিভারি
Exosome is coming out of the cells. So they are like nanoparticle mediated drug delivery people do. Exogenous nanoparticle mediated drug delivery. Yeah. So those are actually targeting.
Liposome
बोलते जाइए. Liposome. And that is liposome and this is exosome. Exosome is coming out of the cell. We can detect based on exosomal cancer.
exosome to body cellular part.
হ্যাঁ.
আর কেউ প্রশ্ন করলে করো।
এই তো করবে তো কে হ্যাঁ ওটা একটু বলতে হবে বেশি বলতে হবে
Sir, আমার একটা question.
শিওর
Sir, we know that recent trend in medical science research that is true drug for true person or specific drug for specific.
exactly what I said. Yeah.
So what are the roles of biomarkers in this field?
Yeah. So unless you identify how the individual is different. So what you just said is individualistic drug, precision medicine. So 5 different person having pancreatic cancer or breast cancer, they have different type of mutations. So unless you know how should they change,
Yeah, because
So-
polymorphism is
polymorphism or at the DNA level, at the RNA level, at the protein level, first you need to know কি চেঞ্জেস গুলো আপনি দেখতে চাইছেন সেটা তো আগে দেখতে হবে না কি চেঞ্জেস গুলো আমি এক্সপেক্ট করছি সো ওয়ান্স উই নো হোয়াট আর দ্য চেঞ্জেস উই এক্সপেক্ট দেন দ্য চেঞ্জেস ইটসেলফ বিকাম বায়োমার্কার ওই চেঞ্জেস গুলোই তো তখন বায়োমার্কার হয়ে গেল তাই না
SNP এর ক্ষেত্রে স্যার ইটস ওকে বাট মাল্টিপল যখন polymorphism
না মাল্টিপল তখন তো এসএনপি আর মিউটেশন তো জাস্ট ফ্রিকোয়েন্সি এসএনপি যখন ওটা ফ্রিকোয়েন্সি বেশি তখন এসএনপি কিন্তু যখন ওটা মিউটেশন হয়ে গেল আমি যে কেরাসের মিউটেশন কথা বলছিলাম দ্যাটস লেস দ্যান 1%, দ্যাটস মিউটেশন। তারপর ধরুন ট্রান্সক্রিপ্টোমে যখন যাচ্ছেন জিন এক্সপ্রেশনের লেভেল কপি নাম্বার অফ পার্টিকুলার এমআরএনএ, প্রোটিনে আসছেন যখন কোন প্রোটিনটা বেশি সেইটা, মানে অ্যাট এভরি লেভেল যেইমাত্র আপনি আইডেন্টিফাই করলেন হোয়াট আর দ্য চেঞ্জেস those changes are biomarker.
Yeah, sir. My question is here. So if we think about that is true drug for true person.
Yes,
So success rate কি?
সাকসেস রেট ইজ হিউজ। সো আমি একটা দেখালাম না, একটা স্লাইড ছিল ওখানে, ওই চা পরে হবে, না চা পরে হবে। এই যে এখানে একটা স্লাইড ছিল, এই স্লাইডটা। So you see, like based on all these mutations, see how many different drugs are already designed এবং they are under clinical trial. এই যে নাম্বার দেওয়া যেগুলো, সেগুলো হচ্ছে ক্লিনিক্যাল ট্রায়ালে আছে। এটা হয়তো এই স্লাইডটা আমি নিয়েছি 1-1.5 বছর আগে, এর মধ্যে হয়তো কেউ ড্রাগও হয়ে গেছে। So what I'm saying is, 3টে স্টেপ, you have to identify what are the changes. সেই চেঞ্জেসগুলো কিন্তু বায়োমার্কার হয়ে গেল। তারপরে তার উপর বেস করে ইউ আইডেন্টিফাই পার্সন 1, পার্সন 2, পার্সন 3 হাউ দে আর বিহেভিং। তখন সেই চেঞ্জেসের এগেনস্টে আপনি ড্রাগ ডিজাইন করছেন। তার মানে পার্সন 1 এর জন্য একটা ড্রাগ, পার্সন 2 এর জন্য একটা ড্রাগ, পার্সন 3 এর ট্রু ড্রাগ ফর 2%।
Thank you, sir.
So what you are saying is very important, because when my gastroenterologist collaborator, they told, why don't you look at the microbiome? So actually there's a huge role of microbiome, how they are processing the foods, the metabolites, and because most of the time they are those metabolites produced by microbes, they are related directly to immune system, because they are being absorbed and they modulate the host immunity. And chronic inflammation bolun ba cancer bolun, it's just all interlinked.
In true sense predisposing factorটা ওইটাই, মানে যদি আমি
Absolutely, completely.
করার আগে আমি যদি ফুড হ্যাবিট থেকে মাইক্রোবায়োটাটাকে ধরে ফেলি, যে
It's all linked.
means this,
And again, so it's host genetics and microbiome. They cross talk together. মানে কিভাবে তারা, so that's why we need a comprehensive view.
ছোট একটা জিনিস একটু এই প্রসঙ্গে বলি, সেটা হলো গিয়ে দেখা যাচ্ছে সার্ভাইকাল ক্যান্সারের ক্ষেত্রে মাইক্রোবায়োটা কিন্তু মোটামুটি কনসিস্টেন্ট, কিন্তু সার্ভাইকাল ক্যান্সারের মতো না, নর্মাল সার্ভিসের ক্ষেত্রে কনসিস্টেন্ট মাইক্রোবায়োটা থাকছে, কিন্তু যেই ক্যান্সার ডেভেলপ করছে
চেঞ্জ হয়ে যাচ্ছে,
প্রোফাইল চেঞ্জ, যেটা কিনা আমাদের গার্ডি আবার ডিসবায়োসিসটা অনেকটা বেশি।
উল্টোটা বলছো চেঞ্জ হচ্ছে বলে ক্যান্সারটাও
ক্যান্সারটা হচ্ছে,
এটা ভেরি ডিফিকাল্ট টু ধোয়া, এটা বলাটা খুব ডিফিকাল্ট যে হু ইজ দ্য কজ এন্ড হু ইজ দ্য এফেক্ট, বাট
সেটা
বলাটা ডিফিকাল্ট, বাট নাও পিপল আর
How come is the change coming on? I mean, suddenly or
টাইম
No,
কোর্স করলে ধরা
এটা খুব প্রচন্ড কোরিলেটেড না, মানে কোনটা আগে হচ্ছে। সো ইজ ইট বিকজ অফ প্যানক্রিয়াটিক ক্যান্সার দ্য গাট মাইক্রোবায়োম ইজ চেঞ্জিং, অর বিকজ অফ দ্য চেঞ্জড গাট মাইক্রোবায়োম? সো স্টেপ ওয়াইজ যেতে হবে। ফার্স্ট এস্টাবলিশ করতে হবে whether there is a change or not. Once you have the change, then you go back to your cell culture lab and see যে এবার ওই ব্যাকটেরিয়াটা যে মেটাবোলাইটটা প্রডিউস করছে, সেইটা যখন সেলের মধ্যে দেওয়া হচ্ছে, সেটা সেলের মধ্যে প্রলিফারেট করাচ্ছে কিনা।
থার্ড প্লেয়ার চলে আসছে এইচপিভি,
থার্ড পেয়ার চলে আসছে অন্য। ইটস ভেরি ডিফিকাল্ট, বাট লট অফ স্টাডিজ আর বিং ডান। অ্যাকচুয়ালি আমারই একটা প্রজেক্ট স্টার্ট করেছি, কিন্তু ওই ফান্ডেড হয়নি বলে এখনো বেশি এগোনো যাচ্ছে না। সো ডিফারেন্ট স্টেজেস, অ্যাকিউট প্যানক্রিয়াটাইটিস, ক্রনিক প্যানক্রিয়াটাইটিস, যারা আবার রেকারেন্ট অ্যাকিউট, সো রেকারেন্ট অ্যাকিউট প্যানক্রিয়াটাইটিস এবং প্যানক্রিয়াটিক ক্যান্সার মেটাস্টাসিস, এই পুরো 5টা ইন্ডিভিজুয়ালের আমি স্টুল স্যাম্পল কালেক্ট করছি। ওটাই অ্যাকচুয়ালি এমস এর সঙ্গে কোলাবোরেশন। সো স্টুল স্যাম্পল কালেক্ট করার পর উইল প্রোফাইল দ্য গাট মাইক্রোবায়োম, অ্যান্ড এখন শুধু গাট মাইক্রোবায়োম প্রোফাইল করে কিছু হবে না, so you have to look at the metabolome also, because that is the clinic main part.
when we are talking about microbiome and when we are talking about the gut microbiome, so we can see that this probiotic that we are actually nowadays we are consuming. Previously we used to consume card and all those things, but now we get ready.
Yeah, ready.
Right? So what does it have this effect? How much it is effective?
Yeah, that's
এই ধরনের স্টাডি তো করতেই হবে।
করতেই হবে। সো অ্যাকচুয়ালি এগুলো তো মানে ওই একদম ওই স্পেসিফিক ট্রায়াল করতে হবে না, আনলেস এমনিতেই এতটা কমপ্লিকেটেড।
তো এসে গেছে না, মানে
ফুড হ্যাবিটের সাথে তো গাট মাইক্রোবায়োম
Yeah, that,
obviously কিন্তু এখন এই আমি একটা এক্সাম্পল দিচ্ছি যে ইউকুড যেটা খুব অ্যাড আমি জাস্ট এক্সাম্পল দিচ্ছি। তো এরকম বা এই যে ও বলবো গাট এই যে ট্যাবলেট, then do you think that this has got some effect?
definitely they might have effect. Definitely they might have effect.
Is
So
it clinically proven or something?
জানি না এবার এগুলো যখন ইউজ হচ্ছে তার আগে কিরকম ক্লিনিক্যাল ট্রায়াল হয়েছিল কিনা জানি না।
We used to depend on God. বাড়িতে দই হতো এবং আগে
বাট আমার মনে হয়
আমরা যে এখন যে কার্ডগুলো যে আমুল থেকে নিয়ে
আমুল,
আসছি
exactly.
ল্যাক্টোব্যাসিলাস পাচ্ছি না আমরা যে স্টেনিং করে প্র্যাকটিস করব
আমি
ল্যাক্টোব্যাসিলাস
হচ্ছে বাড়িতে যখন আমাদের
বাড়িতে পাতা I think we should go back to that. We should go back to that. তোমার টপিক চলে এসো, তুমি চলে এসো না, হবে না। থ্যাংক ইউ।